So glycolysis is necessary because it helps us get the energy we need from the food we eat. Glycolysis can happen whether oxygen is present or not present, which is called anaerobic and aerobic, but that is discussed in a different video. The anaerobic processes are discussed in another video. For now, we're going to talk about glycolysis, which can be broken down into two phases, the investment phase and payoff phase. So for this video, we're just going to talk about the investment phase. So here's our happy little glucose. And that's just the molecular structure that it looks like. And glucose is converted into glucose 6-phosphate by hexokinase. And hexokinase takes a phosphate from ATP and adds it onto ADP. So after that process, we will then have glucose 6-phosphate. And glucose 6-phosphate basically is the happy glucose from before, but it has a phosphate group added onto it. Glucose 6-phosphate is converted into fructose 6-phosphate by phosphoglucoisomerase. And that basically changes the shape of glucose from the hexagonal shape to the pentagonal shape. So from what it looks like a 6-ring or a 6-carbon ring structure takes into a 5-carbon ring structure. Then, so with fructose 6-phosphate, it's converted by one of the most regulated enzymes in glycolysis, phosphofructokinase. Some people call it PFK1, but it's most regulated because it catalyzes the first unique step in glycolysis, converting fructose 6-phosphate to fructose 1,6-bisphosphate. And I apologize, I forgot to add in that it takes a phosphate group from ATP and adds that onto the fructose 6-bisphosphate. So ATP is converted into ADP from this reaction. But it's understood that it's in the process. I just forgot to add it, and I just forgot it until after I edited the video. So fructose 6-phosphate, let's go back. This is what the structure looks like. You see how it changed from what glucose 6-phosphate looked like to this structure, which is fructose 6-phosphate. So after phosphofructokinase uses that phosphate from ATP I mentioned earlier, it changes fructose 6-phosphate to fructose 1,6-bisphosphate, and that now it has 2 phosphate groups added onto its structure. So after this, aldolase, an enzyme called aldolase, separates this molecule into 2 different molecules. I'm so sorry, that's my child in the background. But it separates into 2 different molecules. One of them is called G3P, glycerolaldehyde 3-phosphate, and the other one is DHAP, dihydroxyacetone phosphate. Please forgive me, my tongue is tied. But you have these 2 structures that aldolase produces after separating fructose 1,6-bisphosphate, and glycolysis needs G3P in order to move forward to the next phase. Well, how do we convert DHAP to G3P? Well, there's this fun enzyme called isomerase. And isomerase, as it will be shown in here, isomerase will convert DHAP to G3P and vice versa. Isomerase can also convert G3P to DHAP. DHAP we're not going to talk about in this video because that's used to formulate triglycerides, but for the sake of the mention of that, you can definitely go look at another video, look at my other video on that. But for the sake of this video, I'm going to put isomerase right here so that you can better see how DHAP is converted to G3P. And as I mentioned before, G3P is used in order to move forward with glycolysis to the next phase called the payoff phase. So that is the next video after this. So after you watch the investment phase, please go check out the payoff phase of glycolysis. I want to thank you guys so much for watching my little video on glycolysis part 1 investment phase. See you in the next one. Bye. Bye.